All right, let's take a look at the last category of ways in which insects can protect themselves once these plant secondary compounds have actually reached their bodies. Once the tissue is consumed, there's really two different options of dealing with it. One is to get rid of that compound as quickly as possible. And this happens through a process of detoxification, making them less toxic, and excretion, making them water soluble so that they can go out with the waste fluids. And the other is it comes in and you package it up and put it into tissues that actually are not sensitive to damage. You sequester them. You put them aside without getting them out of your body. So let's take a look at these in turn. This actually is a really nice schematic that kind of outlines the various ways in which you can think about the ways in which allelochemicals, or these plant secondary metabolites, can come in. They first have to come into contact with insect tissue. This might be in the midgut. Sometimes it's actually on the surface of the cuticle itself. They have to actually enter the body, the internal part of the insect, and that basically means going in through the gut and poking in. It basically means going into the gut and then penetrating from the gut into the body cavity, into the hemocoel. Once they've reached this point, a couple of different things can happen, as I've mentioned. One is that the metabolism of the insect can actually break down and excrete these compounds. The other one is that they're packaged away into tissues that don't matter. And the other one is that the reason why these plant secondary compounds are toxic in the first place is that they interfere with some key physiological process that the insect is trying to do, whether it's neural signaling, or whether it's a key digestive function, or whether it's replication of the cell, or something like that. And the reason why these compounds work is because they are a chemical structure that will bind usually to a protein of some kind. And that protein, once it's bound up, has these downstream consequences. It either stops working because it's locked in like that, or it causes it to malfunction and create those problems for the insect. Well, if that protein is just in the slightly different shape so that plant secondary compound can't fit anymore, then you are relatively immune to the effect of that compound. It's inert as far as the insect is concerned, as long as that target is no longer sensitive. So this is sometimes referred to as target site insensitivity. So where the target of that molecule that the plant has created through mutation is of a different shape, so that it's no longer really a problem. So this is a third way in which insects can become resistant to these secondary compounds. You can't really read this very well, but maybe if you zoom in, the short of it is that what this table is trying to show you is that every single one of those mechanisms exists in insects. All of them have been documented. They've been documented for different. They've been documented to work for different plant secondary compounds by different insects and so on. So yes, there's a lot of different ways in which insects can do this. Let's take one of these as an example, because it is a very well studied one. And one is the detoxification and then excretion of these molecules. One of the reasons why these plant secondary metabolites can often be so problematic is that they can be fat soluble. And a lot of the membranes of insect cells and of the neurons in particular are basically lipids. They're the classic phospholipid bilayer of that make the structure of cells. So because these compounds are lipophilic, they can enter cells through these fatty membranes. If you can make these compounds less lipophilic by making them water soluble, then they can't enter as easily and they can be excreted. One of the molecules that does this within the body of insects is a group of enzymes called mixed function oxidases, MFOs. And one group of mixed function oxidases is the cytochrome P450s. And all you need to know about these is that they are a group of membrane-bound proteins, often in the midgut. And what they do, because they're oxygenases, they will take oxygen from the environment that the hemocele is in. They will take the substrate, which is lipophilic here, and they will basically combine and put a hydroxyl group. And OH groups tend to make these molecules more water soluble. And in doing so, they have the capacity not to enter cells and to be excreted. And so this is a classic way in which many insects have developed ways of taking these lipid-soluble substances and making them less toxic, is through these mixed-function oxidases. Mixed-function oxidases, cytochrome P450s, are often inducible. That is, when these compounds start to come into the body of an insect, there's a cascade of signaling that basically alerts the physiology of the insect that, we're in trouble. There's these compounds here, and when these compounds are present, it's time to start upregulating the concentration of these molecules that will put these hydroxyl groups on the end of these toxins. And here's an example of what happens with this cytochrome P450 activity that is sensitive to nicotine for tobacco hornworm. Tobacco hornworm is, as the name implies, actually can feed on tobacco, but it does so by upregulating these cytochrome P450s to then get rid of the nicotine. And here you can see kind of the upregulation, which is dose dependent here. The more nicotine you put in them, the more of these enzymes are produced, and therefore the more they're able to excrete them. And the caterpillars that are reared on nicotine in the first place are less sensitive when they re-encounter nicotine subsequently. So here's an example of a caterpillar that's never been fed on that's been fed on a diet that doesn't have this, and one that has been fed on nicotine. And the ones that have been fed on nicotine and then are injected with nicotine subsequently are able to kind of come back to their senses more rapidly and don't experience these classic kind of physiological twitching that is characteristic of kind of an impact on the neural system, compared to the ones that had not been previously exposed to the nicotine and have not had the chance to upregulate those cytochrome P450s. Those take a much longer time to actually get back to normal, almost twice as long, and show evidence of that physiological response, physiological response to them. Another way in which insects deal with these toxic compounds is to package them up and put them into tissues that are less metabolically active. Classic case for this is in butterflies that feed on toxic plants. Some of these toxic secondary plant compounds are put into the wing tissues. These are generally metabolically inert tissues that are not sensitive, that is not where the important physiology is happening: digestion, growth, reproduction, etc. They're basically a storage area. Now, the advantage of doing this is that if there's anything that tries to feed on the butterflies, they will encounter these secondary compounds just like the caterpillars did when they were feeding on the plants originally. And you've seen this picture already in Amy's talk, where vertebrate predators that are sensitive to cardenolides, these are called heart poisons sometimes because of the effects that they have on the neural system, will find them very distasteful, will throw them up, and then they will actually learn to avoid them in the future. And the basic story here with insecticide, with resistance, actually mirrors very closely something that we see with insecticide resistance as well. You can think of insecticides as human manufactured or sometimes plant derived compounds that act on the physiology of the insect to kill them, to otherwise disable them. And just like insects feeding on plants, pesticides or insecticides particularly have to be encountered by the insect. That encounter can happen through the cuticle or through the guts, and then has to find its way to the target where they will have these negative effects and kill insects. Well, the insects, under very high selective pressure, because if you can't deal with this, you're going to die, are under very strong pressure, selective pressure, to actually select for traits that allow them to be able to deal with this. Those insects that have those are going to become more common in the population compared to the ones that don't. And these include things like reducing the likelihood that these chemicals actually enter the body in the first place. So allelochemical avoidance mechanisms such as behavior or sequestration. This is also a way in which insects dealing with insecticides can similarly deal with them. Once they have actually entered the body, you can have detoxification enzymes that are selected for to become more efficient at breaking down or excreting those. And you can also have kind of target site insensitivity at the end here. So everything from detoxification, avoidance, and insensitivity that is present in the way in which insects deal with plants also come to bear when insects are dealing with insecticides. And the more infrequent the exposure is to either plant chemicals or to insecticides, the more likely you are going to be using these sorts of mechanisms. Whereas if the likelihood of encounter of these chemicals is very high, then you probably will have a series of traits that allow you to do this, including these target site insensitivity. But this has to be under very high selective pressure. Here's an example from one of our own, Michael Crossley, former student here in our department, that actually looked at the number of genes that were responsible for detoxification of one insecticide here, imidacloprid. This is a neonicotinoid. As you can guess from the name, it has similar attributes as nicotine has on the nervous system. The Colorado potato beetle, Leptinotarsa disseminata, is one of the most damaging pests of potatoes and of other solanaceous crops. And through its evolutionary history with the Solanaceae, it's been dealing with all kinds of these nasty nitrogen-containing toxins throughout its millennia of interactions. And therefore, it's evolved a series of traits encoded by these genes that allow them to deal with not only nicotine, but also with the neonicotinoids such as imidacloprid. And one of them actually has to do with, some of these are expressed actually in the cuticle, others are in the cells themselves. Here's the cytochrome P450s, CYP genes here. There's another class that I didn't even talk about, which are glutathione S-transferase genes, which get rid of sulfur-containing molecules. And here's traits for target site insensitivity. So this is all to say that insects are not just passive partners in this relationship with plants, but they have the capacity to actually respond to and evolve resistance and traits that allow them to deal with these plant secondary compounds. And this really sets the stage for these reciprocal interactions between plants and herbivores.